So let me walk you through our Drug Hunter webpage. So the webpage that you see here, it's very user-friendly. On the top right, you can log in with your email address or your phone number. You see what institution you're belonging to. Yeah, it's a very, very simple passwordless access to the page. One great place to start if you haven't used Drug Hunter before is our molecule flashcard that you can use for some inspiration. Every time you refresh the page, a new molecule will pop up with the case study, and you can consume this on a quick coffee break to, maybe use some transferable insights for your project. On the right-hand side here is our homepage, so you will see some of the recent articles that we've published from different categories, and I will walk you through each of these categories momentarily. You can also click on this latest tab here where we have a beautiful card overview, so you can just simply have a better overview in a different browsing format to quickly see what's important and what you need to pay attention to. Another great place to start is our most popular tab here. If you want to see what other people are reading about, we just recently did a roundup of the 12 most popular industry case studies in 2023. So this, content that most of our users find very valuable. We also have resources such as a practical PK calculator that you can use to plan your PK study, and that also helps with interpreting results. By far, our most popular type of case studies are industry case studies. So I will just, for example, select this one here. This is a first-in-class amino acid transporter inhibitor from Jnana that was discovered by reactive affinity probe screening, and it's for the treatment of PKU. So in those types of articles, in this case, it's from a first disclosure, so that's not even published here. We will first give an overview of why is this molecule important or this target important, and why do you need to pay attention to it. So here we have a first-in-class treatment for PKU. And this is essentially really nice because Jnana just recently announced positive proof of concept data with significant phenylalanine reduction in the blood, which is really good news for patients because they don't have any other small molecule options for PKU currently, as the current treatment is more dietary restrictions. We also talked briefly about the indication. The indication here, PKU, as I mentioned, and PKU is caused by a disruption in the PAH gene that's then leading to alanine accumulation in the brain, which could then potentially lead to severe cognitive impairments. And you get really quick one paragraph overview of the biology, and you don't really have to go through a 20-page Nature review article in order to grasp the concept. Again, we talk a little bit about the target. The target here is a solute carrier protein that is responsible for the intestinal absorption and the renal reabsorption of dietary amino acids such as phenylalanine. And for the longest time, this was really deemed undruggable, but Jnana used a new approach here that we're describing briefly, also for you to quickly understand and grasp the concept. This concept here is a rapid affinity probe screening that was pioneered by Ben Kravatz and Chris Parker's lab in San Diego. And that really allowed the team to identify a starting point that would not have been discovered in traditional high throughput screening. We also briefly explained the concept of photoaffinity labeling, quickly giving you an overview of the involved chemistry and the involved biology. If you do want to read more about this, we link all these purple sources out here that lead to the original references, and we include anywhere from 80 to 100 references in each individual article. And we don't only link other academic journal articles, we also link conference presentations, press releases, labels, really all information that's relevant to you. The medicinal chemists in the audience, we will also highlight some key points in the SAR that will allow you to focus immediately on the key problem. So we highlight this with different colors. You don't have to go through many different publications or one table. That we quickly drive home the points of what the problems that were addressed in this case here: CYP inhibition, hERG inhibition, and also hepatic clearance problems that were all optimized in the final candidate. The clearance problem here, as you can see, very nicely resolved by changing that aromatic piece here on the right hand side of the molecule. For compounds that are at the clinical development stage, we will also put in some clinical information to just briefly give you an overview of any interesting pharmacology or toxicology that did occur during those clinical studies. We'll piece together the synthesis. In this case, as I mentioned, is from first disclosure. So we piece together the patented synthesis that will quickly help you understand if you want to include any of those pieces or functional groups in your molecule, so you get some synthetic inspiration and get an idea of what kinds of yields you can be expecting. So, when possible, we will also reach out to experts in the field or inventors. So, this particular article was actually reviewed by Dean Brown of Jnana Therapeutics. So, that gave us confidence that we can publish this article. I would argue that these types of articles would significantly accelerate your learning. And by going through one case study a day, you would absorb about 200 to 300 case studies a year. And even with one a week, you can absorb knowledge of 50 different programs that would be orders of magnitude more than from your quarterly journal club. And you'll likely find something useful within kind of the first few weeks of getting into the habit of reading one of these studies on a regular basis. Throughout your tenure in the pharmaceutical industry, you will likely encounter many different projects, and maintaining a broad yet informed perspective on the industry's pulse is not just beneficial, it is essential. Your company probably has multiple different projects in various disease areas, and being able to talk knowledgeably and actively with your colleagues is really valuable if you would like to make suggestions on other projects and become known as a leader in your company. This is where Drug Hunter articles help you form an informed opinion or perspective. Each month, we will be publishing the molecules of the month, so you can kind of see what the industry paid attention to in any given month. It will give you a poster of the 10 top molecules with the executive summary right here, and we'll also link out to the articles that covered them. In some of the cases, we also wrote case studies on them. Then we'll link out to those. So that's a very, very useful resource that our readers find very valuable. A format that we've been experimenting with is roundups of molecules, news, and also most recently patents. So I'm going to focus on patents in this case. So, here you can see a few patents, for example, Denali's LERK inhibitor or a STING antagonist from Roche, one of the NaV1.8 compounds from GSK, really hot topic now with the recent news from Vertex. So in my experience, really sifting through patents takes a long period of time, and you literally have PDFs with hundreds and hundreds of pages that it's very difficult to get information from. Here we just highlight the 12 most interesting patents from December, but we also have an annotated searchable list here. So if you were interested in a specific target, let's say you were interested in KRAS, then you can just quickly search the table and you can see what was the patent activity on KRAS here in the month of December. So that is a really very useful type of format. If you would want to get an overview of certain fundamental topics, for example, how can you get drugs into the brain, or how do degraders differentiate from traditional small molecules, our educational resources here will help you, jumpstart your reading, or make some recommendations for newer hires or starters in the drug discovery field. If you're interested in a specific topic, we also have a very nice dynamic search function. So, for example, you can use this to search for targets such as BTK. If you remember, you were looking for a reversible BTK inhibitor, you can use that, right? And it's dynamically, as I type, will update the search. this is particularly useful for our case studies. We have about 550 case studies out to date, and we're publishing more and more of those. So if you ever see a certain tox phenomenon, like a bleeding phenomenon, occurring in your tox study, you want to quickly understand, like, have other people seen this before? This bleeding risk, for example, here on the TRP1A inhibitor for Genentech was very nicely understood and solved. This was is actually going through an aldehyde oxidase mediated metabolism that makes the molecule look like warfarin and then inhibit the vitamin K redox cycle. That was very nicely solved and addressed here in this case study. But you really wouldn't be knowing about this if you wouldn't follow that specific paper. So if you're one more thing that I'll show you, if you're heading to a design meeting and you just want to do a quick sanity check on how many people put an azetidine in their molecule lately, you just want to see, have people use this, and you type this in there and you get a quick overview of like 40, 50 compounds. So you see you're not crazy by proposing that. But if you would do that on something like SciFinder, you would be just swamped with results and would be less useful. You can also test like acetylene. Have acetylenes been tested lately? And you see there you get a list of about 10 compounds. So you can feel much better about proposing that, and also jumpstart your reading to see how this impacted the properties of the molecules here. So with that, thank you so much for your attention. Please let us know if you have any feedbacks or questions. We're always looking to ways to make our content more engaging and better. Thank you so much for your attention, and hope you find this very useful.